Dear students, the topic is activity of salivary amylase or TALIN in relation to temperature and calculation of temperature coefficient. To determine the rate of activity of salivary amylase or TALIN on the digestion of starch with the effect of temperature using Schooler's sugar titration method and calculation of temperature coefficient Q10. Let we can see the principle of this reaction. In this method, the velocity of enzyme reaction is measured by determining the rate of disappearance of the substrate starch or rate of formation of the end product glucose. Starch undergo digestion and form glucose. The changes in temperature have a marked effect upon the activities of living organisms. The temperature coefficient of a process is the ratio of its speed or velocity constant at a given temperature. That is 10 degrees Celsius is the lower one. That it is otherwise called as Q10. If we know the speed at any two temperatures, that is temperature T1 and T2, we can calculate the temperature coefficient. Have a look on the materials, reagents, calculation of Q10 for this experiment in detail. The materials used for this experiment are test tubes, test tube stand, pipette, measuring jar, burette, conical flask, incubator, etc. Enzyme amylase preparation. Rinse the mouth with water, collect the saliva in a beaker. Dilute the saliva to a non-concentration, that is 1:20, and filter through a glass wool. Buffer solution. Powder the BDH buffer tablet of pH 6.8 dissolved in 100 ml of distilled water. Substrate preparation. The substrate is starch. Dissolve 1 gram of starch powder in 100 ml of distilled water. Boil and cool down. Then filter through a glass wool. Sodium chloride. Dissolve 1 gram of sodium chloride in 100 ml of distilled water. Fillings A solution. Dissolve 34.65 gram of copper sulfate in 500 ml of distilled water. Fillings B. Dissolve 174 gram of sodium potassium tartrate and 125 gram of potassium hydroxide in 500 ml of distilled water. Potassium iodide solution. Dissolve 30 gram of potassium iodide in distilled water and made the solution up to 100 ml. Let we can see the procedure. Take 5 clean test tubes, mark it as A, B, C, D and E. Into each test tube, add 3 ml of saliva, 1 ml of substrate, 1 ml of sodium chloride and 1 ml of buffer solution. pH should be maintained at 6.8. Then incubate these 5 test tubes at 5 different temperatures, that is 10 degrees Celsius, 20 degree, 30 degree, 40 degree and 50 degree Celsius respectively for 2 hours. After 2 hours of incubation, take 5 to 50 ml Corning conical flask, into which add 10 ml of fillings A, 10 ml of fillings B, 5 ml of incubated mixture and 25 ml of distilled water. Mix well the contents and boil exactly for 3 minutes first, then continue another 2 minutes. After boiling first 3 minutes and next 2 minutes, cool down the contents rapidly under running water until lukewarm. Then add 10 ml of 30% potassium iodide and 10 ml of 30% sulfuric acid. Add 0.5 ml of starch as indicator. Color will develop as blue, dark green or dark yellow. Titrate with 0.1 normal sodium thiosulfate taken in the burette. Endpoint is the disappearance of color. This table shows the summary of the experiment. This is the calculation for the enzyme activity for 1 milligram of glucose in 60 minutes. First, volume of sodium thiosulfate used at 10 degrees Celsius is 4.2 ml. Milligram of glucose present in 4.2 ml that is equal to 0.4 into 2, that is 0.8. So from Schooler's sugar titration table, we got 81.8 minus 0.8 is equal to 81 milligram. Hence, enzyme activity for 1 milligram glucose in 60 minutes is equal to 81 divided by 60 is equal to 1.35. This is for 10 degree Celsius. Like this, we should calculate for 20 degree, 30 degree, 40 degree, 50 degree. Next part is calculation of temperature coefficient, that is Q10. Q10 is equal to K1 divided by K2 into 10 divided by T1 minus T2. What is K1? Rate of activity at temperature T1. K2 rate of enzyme activity at temperature T2. You know T1 is higher temperature, T2 is the lower temperature. Here is the calculation of Q10. The value is 1.029 for 10 degree and 20 degree, 0.966 for 20 and 30, 1.009 for 30 and 40 degree, then 1.029 for 40 and 50 degree Celsius. This is the glucose conversion table from Schooler's titration method. Dear students, I hope you can do this experiment in your laboratory. Thank you for watching. See you in next class with another topic.